पीस ले लो। 5 पीस तो कम से कम खाएगा सर।
other than hydrogen. Other than those that transfer hydrogen are NAD, FAD, FMN, etc. One example of transfer of FAD, one example of transfer of NAD. Transfer of pyruvate being converted into lactate. Or we can say being converted, this reaction is reversible. We can say pyruvate being converted into lactate or lactate being converted into pyruvate. In either ways we can say. So when we say pyruvate being converted into lactate, is happening. NADH is being converted. This reaction is reversible. Pyruvate is being converted into lactate. This reaction is reversible. In this way, pyruvate being converted into lactate. What is happening? NADH is being converted into NAD. This NAD is used in the glycolysis. This NAD is consumed in the glycolysis pathway and it is used for the formation of energy, and it is used for the formation of energy. So NADH and H is converted. This NAD is used in the glycolysis pathway for the formation of energy. So in this we can see what is happening. Pyruvate is being reduced, pyruvate is being here there will be removal of hydrogen, so NAD is being oxidized and pyruvate is being reduced to lactate. And the formation of NAD is very important because it is more important than that of formation of lactate, because this enters into the glycolytic pathway and leads to the formation of energy.
साला कहाँ पर आ रही है?
In the other way, we can say, we take it in the other way, we can say the lactate is being, the substrate lactate is being oxidized, and in the other way, the lactate is being oxidized and NAD is being reduced. This NAD is a coenzyme which is synthesized from nicotinamide, which is a member of the B complex group. And as I told you, this reaction is reversible, lactate to pyruvate, pyruvate to lactate. And the enzyme is catalyzed by lactate dehydrogenase. Hydrogen molecules are removed, out of which one hydrogen and two electrons are accepted by NAD. One hydrogen and two electrons are accepted by NAD. Form NADH and H, and the remaining H is released into the surrounding medium. So do you understand this reaction? I think you have not read the glycolytic pathway. When you will read the glycolytic pathway, you will understand much better. So the first group, classification of coenzymes, the first group is the transfer of the hydrogen atoms. In the second group, so the first two examples are NAD, FAD, FMN. In the second group of coenzymes, it is the transfer of groups other than hydrogen. Transfer of groups other than hydrogen. What happens in this? A particular group or radical is transferred from the substrate to another substrate or from the coenzyme to the substrate. Here, the enzymes are also considered as co-substrate. So, the examples of the second group are TPP, that is thymine pyrophosphate, and the group transferred is hydroxyethyl, and the group transferred is hydroxyethyl. Another example, pyridoxal phosphate, PLP, which you must have read in, you haven't read amino acids. You will be reading in the reaction of transamination. So it transfers the amino group. It transfers the amino group. PLP, it transfers the amino group. Then other examples are biotin, which transfers carbon dioxide, the coenzyme A, which transfers the acyl group, the tetrahydrofolate, which transfers the one carbon groups. So, a few examples. So, TPP, which transfers the hydroxyethyl group, pyridoxal phosphate, which transfers the amino group, biotin, which transfers carbon dioxide, coenzyme A, which transfers acyl groups, and tetrahydrofolate, which transfers one carbon group. So these are the examples of the second group of coenzymes. First group of coenzymes transferring the hydrogen, examples being NAD, FAD, FMN. The second group of coenzymes, the second class of coenzymes, which transfers groups other than the hydrogen groups. In this we have many examples. TPP,
तो बोलती है कि नाली को उठा के कहाँ ले जाए, जब नाली को उठा के ले जाने के लिए बोल रहे हैं, हम बोल रहे हैं सोल्यूशन कर रहे हैं, सोल्यूशन बता रहे हैं, तो आप बोलते हैं झगड़ा करने के लिए आप सोचते हैं, झगड़ा बात बोलते हैं, तो उसका बेटा आ गया, मतलब बोलने का तमीज़ है, बोलेगा तो क्या बोले हैं, बोलने का तमीज़ है.
The mode of actions of enzymes, or we can say the mechanism of enzyme activity, how the enzyme acts.
तुम किसी को सुनाएगा, तुम नहीं सुनेगा.
Important thing is the lowering of activation energy.
सोल्यूशन करने को मतलब लोग हम कुछ भी कर सोल्यूशन कहाँ हमको देगा.
Whenever a reaction takes place between the substrates, in the non-enzyme catalyzed reaction, what is happening? The substrates, they combine, they first go from the ground level to a transition state in which they have in the transition state they have the energy, the free energy in them is the maximum, and then they go on to the formation of the product. But what happens when there is in an enzyme catalyzed reaction, there is graph. If we draw a graph, reaction time. We draw a graph, free energy and the reaction time. The non-enzyme catalyzed reaction goes this way. Suppose we have substrates A and B, they go into a transition state. The highest maximum free energy, suppose the free energy is 0 here, the maximum free energy will be at AB, this is the transition state, and it goes into the formation of C and D, the products. It goes into the formation of the product C and D. Now what happens is that in the enzyme catalyzed reaction, enzyme catalyzed reaction, there is lowering of activation energy. This is the activation energy. This is the activation energy. Non-enzyme catalyzed reaction. This is the enzyme catalyzed reaction. And this is the activation energy. So what is happening in the enzyme catalyzed reaction? There is lowering of the activation energy. There is lowering of the activation energy. Lowering of the activation. And the change energy. Suppose the free energy is G0. This is the delta G0, which is the change in the free energy. The change in the free energy in both the enzyme catalyzed reactions and the non-enzyme catalyzed reaction. The change in the free energy is the same. Only it's lowering of the activation energy.
क्या बोल रहे थे पापा?
You can see this is the enzyme catalyzed reaction and this is the non-enzyme catalyzed reaction. So first the substrates go into a transition state in which they have there is an increase in the free energy. The ground level, much increase in the free energy, the activation energy. So what is activation energy? Activation energy is defined
तुमने कुछ बोला था क्या?
as the energy required to convert all molecules in one mode of reaction. All molecules in one mode of the reacting substance from the ground state to the transition state. From the ground state to the transition state. Activation energy is defined as the energy required to convert all molecules in one mode of reaction, in one mode of reacting substance from ground state to the transition state. And what we can see is that enzymes reduce the magnitude of this enzyme activation energy. Now coming to the Michaelis-Menten theory.
क्या बोले वो क्या बोलते हैं वो हम लोग का बात बताने लग गई ना.
वो सब बोलने लगी इसलिए ना जब नाला का हो रहा तो वो सब बोल रही है.
Michaelis-Menten, they put forward an enzyme substrate complex theory. The enzyme combines with the substrate to form the
तुम तो सुबह पढ़ गए.
substrate complex, which immediately breaks down to the Michaelis-Menten put forward a theory known as the enzyme substrate complex theory. The enzyme complex, which then immediately breaks down to the enzyme and product. So enzyme and substrate, they combine, form the enzyme substrate complex. This then breaks down to form the
सुबह तुमने क्या किया है?
enzyme and the product.
गदर मचा कर
Now coming to the next, Fischer's template theory. Fischer's template theory. Now, what did Fischer say?
अरे
Fischer's template. According to it, first
बताया तुम्हें तुम्हारे इतना बढ़िया से नहीं होता है कोई फुल डिटेल में.
enzyme
बता थोड़ा निकाल आया. थोड़ा जल्दी बोलो.
the substrates, the enzyme is the substrate. Fischer said
अपना पापा बातें
that
लगा.
the three-dimensional structure of the active site of the enzyme is fixed. Structure of the active site of the enzyme, it is fixed. And the substrate comes in contact with the enzyme, the reactor is them. So this active site of the enzyme is fixed. The active site present in the enzyme is fixed. So the substrate comes in contact with the enzyme, the reaction takes place. Substrate and enzyme and reaction takes place. And this is also known as the lock and the key mechanism. Just as the key fits into the lock, key being the substrate, lock being the enzyme come in contact with each other and the reaction takes place. What Koshnack induced before. Koshnack said that is not fixed. Enzyme not fixed. Active site of the enzyme is not fixed. Substrate. When the substrate comes in contact with the enzyme, what happens? The active site of the enzyme, it undergoes conformational changes and then it undergoes conformational changes and then the reaction between the enzyme and the substrate takes place. So conformational changes are occurring at the active site of the enzyme, concomitant with the combination of the enzyme with the substrate. This is not fixed. When the substrate comes in contact with the enzyme, what is happening? When the substrate is coming in contact with the enzyme, what is happening? Conformational changes are taking place and then the enzyme comes in contact and the reaction takes place. Types of the enzymes are not fixed. So at first the substrate binds to the specific point of the enzyme and then leads to more secondary binding and conformation, that is conformational changes take place and then the reaction takes place. So the substrate induces conformational changes in the enzyme and then the catalytic groups come in contact with the enzyme. The catalytic groups of the enzyme come in contact with the substrate and then the reaction takes place. Types of the active site. What is the active site? The active site of the enzyme, you must know that. Points of clear lowering of the activation energy. Michaelis-Menten theory, which says that the enzyme and the substrate must come in contact with each other, and first the enzyme substrate complex is formed and then it goes on proceeds to for the formation of the enzyme and the product. Product is formed, enzyme is theory said that the active site of the enzyme was fixed. So when the substrate comes into contact with the enzyme, it fits into the enzyme and reaction takes place just as the E fits into the law. The Koshnack-induced fit model, the enzyme, the active site of the enzyme is not fixed. When the substrate comes in contact with the enzyme, it undergoes conformational changes and then the reaction of the enzyme. How is it formed? This active site, you must try to understand this active site of the enzyme. Amino acids which are present at distant points. Amino acids which are present at distant points.
क्या बोले थे क्या लगा लेट बोले थे लाने बाटा कटोरी बोले थे जिसमें रस निकालना
is formed by the coming together of the amino acids present at 5 position and 101. Try to understand. It contains amino acids, and these amino acids are present at distant sites, at distant positions in a polyinostate polypeptide chain. But when they come together, they form the active site. For example, lysozyme, which has 129 lysozyme, which has 129 amino acids.
बड़ा
The
वाला
active
इसमें
site
था
where the amino acid present at the 35th position,
हो जाओगे
the 52 position, the
सस्पेन है और उससे बड़ा कटोरी
63
नहीं
and 101 position. So
इसमें
these
कटोरी
enzymes,
नहीं है बड़ा वाला
these
नहीं
amino acids are coming
थोड़ा
together
सा ले
and
जा
they are forming
इसी में खा लेगा डाल के निकाल
active
दे
sites.
रहा खाली डुबा डुबा के निकालना ही
The
नहीं
active sites, the amino acids, sometimes if serine is increased in numbers, suppose the active site is formed by serine residues, then it is known as a serine comes in the serine
ऐसे
enzyme
करके ना
group.
नीचे मसाला
It is known as serine proteases. For example, chymotrypsin, it comes under the example of serine proteases, means there are a number of serine residues present at the active sites. So, different amino acids may be present at the active sites. This was the active site. Now coming to the factors affecting the enzyme activity. What are the factors that
क्या
नीचे से
the enzyme concentration, the substrate concentration, product concentration, then we have the temperature, the pH, and other substances, some activators, inhibitors, etc. Presence of activators, inhibitors, etc. So coming to each one by one, factors affecting the
बोला
enzyme
प्यार
activity.
थोड़ा सा खाएगा
So we have the enzyme concentration, the substrate concentration, the product concentration. Then we have the effect of the pH, the temperature, and the presence of activators, inhibitors, etc. So coming to the enzyme concentration. Suppose a graph coming to the enzyme concentration. Suppose a graph plotting enzyme concentration against the velocity.
मम्मी आइए गुपचुप खाइएगा
Enzyme concentration against the velocity or the rate of
लेके आया
reaction. Then graph is a straight line. So the velocity of reaction varies proportionately with the enzyme concentration when all the factors, that is the substrate concentration, the pH, the temperature are at optimal level. And this property is made use of in determining the level of a particular enzyme in plasma or serum. Known volume of serum, it is incubated with the substrate for a fixed time and then the reaction is stopped. The whatever product is formed, that is quantitated, and this gives us the estimate of the enzyme, amount of enzyme present in the serum. So enzyme concentration, if a graph is plotted, the velocity against the enzyme concentration, we see that the graph is a straight line. The graph is a straight line. The velocity, that means the velocity of or the rate of reaction varies proportionately to the enzyme concentration, where the substrate concentration, the temperature and the pH are at optimal level. And this property, it is made use for determining the amount of serum present in the sample. Amount of enzyme present in the sample.
हाँ
What we are doing, this is known as air point analysis. What is being done? The known volume of serum is incubated with substrate for a fixed time. The reaction is then stopped. The product, it is quantitated, and the amount of the product is tells about the amount of the enzyme present in the sample. And coming to the substrate concentration. Coming to the substrate concentration. What is happening? What is happening in this?
चोखा घटने वाला है
and S. So the substrate for a typical enzyme, the graph is not a straight line, but it is hyperbola. What happens is that initially, when the substrate concentration is increased, the rate of reaction is directly proportional to the substrate concentration.
लोग पानी एक्स्ट्रा मांगता है, आलू एक्स्ट्रा मांगना चाहिए मैडम। गलती करता है, आलू लोग कम देता है।
Concentration is being increased. Rate of reaction is directly proportional to the substrate concentration.
Hers.
You can see
Hers.
that when the substrate concentration is increased,
सुनान देता है।
there is increase in the rate of reaction.
करना।
Now at B point, what is happening? At B point,
कौन खा के ऐसे
percent
बोले
of the enzymes are reacted with the
यहाँ लगा कर आपको तो
at
क्या
point
दूसरा है?
50% of the enzymes they are reacting with the substrate, and this is the max. So this point is max by
आलू कम दिया है
half the
बताइए।
maximum velocity.
पानी बोला हुआ एक्स्ट्रा
When
ज़्यादा
we draw
दीजिएगा।
a line down
तो
to
बोला
आलू ही बोले थे हम।
get the value of Km.
सब ने इतना कम भर के खाते ही हो
What
घड़ी
is
है।
Km? Km is the
शुरू से ही
substrate
कम भर
concentration
ज़्यादा भर
at
के पता नहीं
which
कहाँ
the
जाता है।
velocity is half the maximum velocity. Km is the substrate concentration at which the velocity is half the maximum velocity.
मम्मी से पूछो रख देंगे, घर में आया ही हो तो रख देंगे।
50% of the enzymes are combined with the substrate, and Km is the substrate concentration.
तो क्या पानी में डालेंगे तो वही होगा ना।
Point C, the maximum velocity is attained. So even on increasing the substrate concentration, there is no rate,
सब्जी कहाँ
there is
बना
no
है?
change in the rate of reaction.
इसे तो पूछ रहा है, क्या को रहने दे?
Substrate concentration, there is no increase in the rate of reaction. So for a typical enzyme, as the substrate concentration increases, is increased, the velocity and increases
बोलते रहे
the Vmax or the maximum value.
वो दुकान
If further increase in the substrate concentration, there is no increase in the velocity.
उसको पूरा नाम लिख से कौन जानेगा सुझाव?
So the shape of the curve,
बता देने का।
hyperbola. Now, Michaelis-Menten equation. What is this Michaelis-Menten?
बुढ़ा लोग का नाम हमको सॉल्यूशन कह देगा। वो क्या करना है करना हम जानेंगे। अभी क्या बोल रहे हैं? बोलो तो कि मुंह खाली कर दूँगा, हाँ कर देंगे। और कुछ नहीं बोला। बोला था हाँ ऐसे ऐसे बोलता है बात बेटा, ये क्या बोला? नहीं बोला कि नाली ऐसी करवा दो, सब साफ करवाइए, नाली का छेद बड़ा करवा दीजिए, बड़ा भी करवा दीजिए।
Km, what is Km? Km is the substrate concentration which the velocity is half the maximum velocity.
मेरा बेटा है, आपका बेटा है,
Michaelis-Menten equation, initial velocity is equal to
कोई बात नहीं किया।
Vmax, that is the maximum velocity, into the substrate concentration by Km plus S.
खाएगा तो खा है लोड गया।
The Km is the Michaelis constant. The Km is the Michaelis constant.
6 को रख दिया मम्मी।
Km is Michaelis constant. Michaelis-Menten equation illustrates a mathematical term. In mathematical term, the relationship between the initial velocity and the substrate concentration. According to the Michaelis theory, the formation of the enzyme substrate complex is reversible, as we already said. And when the substrate combine the enzyme substrate complex and then they form the enzyme and the protein. So this formation of enzyme substrate complex is reversible. And this is K2.
क्या हुआ? क्या टिल्डा?
K2 plus K3 M is equal
वो
to
बोले
K2 plus K3 divided by K1. K2 plus K3 divided by K1. The three different constants can be made into one equation. Which is equal to K2 plus K3 divided by K1. Martelly's mental equation illustrates the mathematical relationship between the initial velocity and the substrate concentration. The Km is the substrate concentration, at which the velocity is half the maximum velocity. So we can also derive this.
कहाँ गया रे?
Is half the maximum velocity.
कहाँ पे क्या है?
Velocity is half the maximum velocity. Speed is max by Km plus S. So we can come Km plus S is equal to Kmax S. So we can say Vmax is equal to Vmax by Vmax equals by Km plus S. So we have Km plus S by Vmax.
खाने बन जा रहा?
Km equal to S minus S equal to Km. At which the velocity is half
मैं कॉलेज में अलग अलग 3 को एड्रेस अब हम देंगे नया एड्रेस
the velocity
क्या पागल है ये
the
लड़की?
maximum velocity. Now coming to the point. The first point A. Here I told you there are 3 points. A, B, C. Suppose at point A, you can say that Km plus S,
सच में बहुत खराब
the
होता है.
substrate concentration is at point A. The graph. At point A in the graph, we can say that the substrate concentration is less than the Km. Substrate concentration is less than the Km at point A. So we can say that Km plus S, then we can say that Km plus S is equal to Km. We can say that Km plus S is equal to K, because the substrate concentration is less than K. So again, putting the values, initial velocity is equal to Vmax S by K. And we can say approximately equal to Vmax. So we can say this initial velocity is equal to this being a constant. S equal to Km. So, taking this value formula, we can say that the initial velocity is almost equal to the substrate concentration, and we can say the initial velocity is the constant is equal to K, the substrate concentration. So we can say the initial velocity is equal to the substrate concentration. Now at point B, where we said that velocity was half the maximum velocity. So at point B, what is happening when the substrate concentration, here we can say that substrate concentration is equal to Km. The substrate concentration is equal to Km. So we can say annotation request क्या होता है? Annotation request. अच्छा चलेगा. Might be that the substrate concentration we can say was point B, there was point A, this point C. So at point B, the substrate concentration is equal to Km. So we can say max S by substituting Km by S, we can say the max substrate concentration by 2S. We can say the velocity equal to the max by Q. Here the velocity is the max by Q. Velocity is the max by Q. So we are saying at point B, the substrate concentration is equal to the Km. So by substituting the value of Km, we can write it in this way and come to the conclusion that the velocity is half the maximum velocity. The velocity is maximum velocity. Now what is happening at point C? Point C. At point C, we are seeing that on increasing the substrate concentration, there is no increase in the rate of reaction. So we can say Km plus S is equal to S plus S. So we can substitute this over there. We can say max S by Km plus S is equal to S. So the initial velocity. So the velocity will be maximum velocity. The velocity at point C will be the maximum velocity. That is, there is no change in the rate of reaction even on increasing the substrate concentration. So we can see the derived and what is the reactions taking place or the rate of reactions at the different points. Again repeating, at point A, it is proportional to the substrate concentration. At point B, the velocity is half the maximum velocity. At point C, the velocity is the maximum velocity. Km is the substrate concentration at maximum velocity that I told you. And does the Km denote? Km denotes, as I told you previously, that 50% of the enzyme molecules are reacted with the substrate at this concentration. At this particular, at that. So 50% molecules are bound with the substrate molecules at that particular substrate concentration. Now, this Km is independent of the enzyme concentration. Km, the few points of Km. Km, you know what it is? And Km, you know that at this point, 50% of the enzymes are reacted with the substrate, and Km, it is independent of the concentration of substrate. And Km puts the affinity of the enzyme to the substrate. If the Km value is high, you should know that the affinity of the enzyme to the substrate is low. So inverse relation, affinity. If the affinity of the enzyme to the substrate. So if the Km is high, that means the affinity of the enzyme to the substrate is low. And if the Km is low, it indicates that the concentration of the affinity of the enzyme to the substrate concentration is concentration. The product concentration is increased. When the product concentration is increased, when the product concentration is increased, it inhibits the reaction, so there is a decrease in the enzyme reaction, or the enzyme reaction is blocked. Then what is the effect of temperature? What is the effect of temperature? Graph like when the temperature is plotted against the velocity. Is bell-shaped. The graph is bell-shaped. So velocity, what is happening? Velocity increases up to a particular optimal temperature. After that, it is decreased. So velocity of enzyme increases with the temperature up to a maximum. After that, which it declines. So it is bell-shaped. The temperature at which the enzyme velocity is maximum is the optimal temperature, which is about 37 to 40 degrees centigrade. For the 50 degrees centigrade, in the human body, it undergoes denaturation. About the temperature, but sometimes the optimal temperature for thermophilic organisms is, which are present in the hot springs, it is 100 degrees centigrade. 100 degrees centigrade. Now, what is the effect of pH? The effect of pH is also to get the graph. In the pH also, the graph is bell-shaped, which means the graph is bell-shaped. pH also, the graph is bell-shaped, which means there is increase in activity with the increase in the pH, and then again the activity decreases. So for different enzymes, there are different optimal pH. So the pH decides the charge on the amino acid residue present at the active site. The optimum pH is 5 to 9. Exceptions for pepsin. Pepsin requires the pH is 1 to 2. For alkaline phosphatase 9 to 10, and for acid phosphatase 4 to 5. Now, presence of activators, inhibitors, and repressors. So in the presence of activators, you will see that there will be an increase in the enzyme activity. In the presence of inhibitors, there is a decrease in the enzyme activity. Depressors and derepressors, again repressors decrease in the enzyme activity. Derepressors increase in the enzyme activity. And there is something as the covalent modification. The process of altering the activity of enzymes by adding or removing groups. That is by making or breaking of covalent bonds is called covalent modifications. If we come to examples of it, covalent modification. One is the phosphorylation and the dephosphorylation. Phosphorylation, some enzymes are active in the phosphorylated forms, some enzymes are inactive in the dephosphorylated forms. So there are many examples in that, and we will come to it. Then the removal of the zymogen form and the zymase form. The zymogen form, which is the inactive form, it is converted into the active form by means of removal of certain groups and by means of removal of certain groups. So it becomes active. Now, so we have completed the factors affecting the enzyme activity. Any problem? Can ask questions? Any problem? No problem? Can ask questions? No problem? So we will continue with this in the next class. And I think we should go into the regulation of enzyme activity, or we will see what we can go into. Any problems? You can ask. So we end the class, children, and in the next tutor we continue with the exam. Yeah.
Thank you, ma'am.
Thank you, ma'am.